Started with our grand rounds. Today I have the difficult job of being Neil Swerdlow, and with apologies for the poor imitation, I need to exhort the trainees to meet with Dr. Nasrallah at 0930 a.m. just across the lobby there. So is that, have I done that okay? Okay. And Henry, as Neil may have told you, that we have a tradition here where the formal introductions of the person's bio and so forth are done by a junior faculty or a senior trainee, which Dr. Lee will do shortly. But I did want to take this opportunity to especially and personally welcome you to the department. Dr. Nasrallah, as you will hear from the formal introduction, actually began his academic career here at UC San Diego and was a very accomplished researcher from the start, got his first grant here, was a very fine teacher, and then lo and behold, he was snatched away from us by kind of a legendary figure in psychiatry, George Winnaker, at Iowa, where he became a head of psychiatry at the Iowa City VA. I think you were only 31 then, weren't you? I mean, that's very precocious, I would say. But anyway, and after that, he's had, as you will hear, a really wonderful career as chair of psychiatry at the Ohio State University and University of St. Louis. Major contributions to schizophrenia, so we'll hear about that. But today I particularly wanted to welcome you as a long-term friend of the department and also to underscore that Henry and his wife Amelia have endowed a lectureship in psychiatry and neuroscience here at UC San Diego. This is going to be a recurrent endowed annual lectureship to bring most distinguished people in the field out to lecture with us about these topics. And so we're very grateful for your thoughtfulness and generosity in establishing the Henry and Amelia Nasrallah Endowed Lectureship in Psychiatry and Neuroscience. And it is fitting that during this 50th anniversary of our department, that this lectureship commences and that you are the first speaker. So I do want to give Henry a round of applause for his talk. Shall we say, the rest of these slackers, they could endow some lectureships too in their topics of interest. What's going on here? Anyway, but seriously now, Dr. Lee, would you like to give a proper introduction to our speaker? Henry, thank you. Thank you.
So it's my distinct pleasure to introduce Dr. Henry Nasrallah, who is the Sidney W. Souers Endowed Chair and Professor of the Department of Psychiatry and Behavioral Neuroscience of the St. Louis University School of Medicine. Dr. Nasrallah is a world-renowned neuropsychiatrist, neuropsychopharmacologist, researcher, and educator. He completed his undergraduate and medical studies at the American University in Beirut. He completed his psychiatry residency at the University of Rochester and a neuroscience research fellowship at the NIMH, where he published 20 papers in 2 years. He was here for, as an assistant professor at UCSD and then went to University of Iowa, where he was an associate and full professor, as well as the youngest chief of psychiatry in the country of the Iowa City VA. And then he assumed the chair position at Ohio State University Department of Psychiatry for 12 years. And during his tenure there, he increased the research funding of the department by 100-fold. And then he served as associate dean and professor of psychiatry and neuroscience at the University of Cincinnati before joining St. Louis University. His research focuses on the neurobiology, psychopharmacology of schizophrenia and mood disorders. He's published over 500 papers and 14 books, including seminal work on the safety and efficacy of medications, cardiometabolic health, and the neurobiology of people with psychotic disorders. He has served as the editor-in-chief of 4 journals, including Schizophrenia Research, Current Psychiatry, and the Journal of Psychotic Disorders, and including the brand new Biomarkers of Neuropsychiatric Disorders. He's the founder of Schizophrenia International Research Society, as well as a distinguished fellow of the American Psychiatric Association, a fellow of the American College of Neuropsychopharmacology, and the American College of Psychiatrists. He has received innumerable awards, including the NAMI Exemplary Psychiatrist Award twice, and many teaching awards, including the USA Teacher of the Year and the Golden Apple Teaching Award. He has been named as one of the world's most cited investigators and has received 95 research grants. In summary, Dr. Nasrallah once responded in an interview in 2011 about what he would like to be remembered as, and he said, a good husband and father, an effective psychiatric physician, teacher, mentor, researcher, editor, and leader, and a fierce advocate for the mentally ill. So Dr. Nasrallah, thank you for joining us today.
Thank you. Thank you very much. Thank you so much for this incredibly warm introduction, and you can tell, I can't tell you how happy I am to be here. Really, this has been a wonderful trip, meeting my old friends. That I started my academic career here, so it's my first academic family. It's like a class reunion in the best sense of the word. But I'm also, I've been following UCSD all the time since I left, and I'm just amazed at what a wonderful institution it has become. It was great when I was here. I didn't realize it was that young when I was here. Only 9 years old. It's now 50 years old. So you are definitely one of the most distinguished universities and medical schools in the world, and I'm delighted to be part of it at one time. And I'm grateful to Lou Judd for recruiting me. At the time, I just so bad again, and haven't seen her in 40 years. In fact, I told her I have a picture. I'm going to start out with first the usual disclosures for CME, and here's the only picture I have of the faculty. Some of you are here already. That I took just before I left to Iowa, knowing that this is my last day or week. So you recognize those people, right? I just saw this guy here. He was talking to me, and who is this? That's right. You guys remember? So here I was, very black at the time, black hair and blue, and with Bob, who's the director of training, Richard Fowler, he's Mark Schacher, David Janowski, Igor Grant, this is Bruce Hubbard there. Remind me the name of the guy. He's here. Oh my God, good to see you. How nice of you to come and share this occasion with me. So this is the only picture I have of the faculty at the time. Have you seen it before? I'd be happy to send it to you if you want. It's a separate email. I'll send it to you. And this is Lou and Pat when they came to our house during my tenure here. It's the only picture I have of Lou and Pat. And then I don't think you can see this very well, but a year after I started here, the residents honored me with Teacher of the Year award. So it was delightful. In fact, that's why I left NIMH, even though I love doing research full time, but I missed teaching and supervising and mentoring residents. And that's why I came here, and it showed in a way. So let's get going. A lot has happened in schizophrenia over the past few years. Maybe let me tell you something interesting. I was just rummaging through some old boxes in my basement the other day and found the abstract of new research at the APA. Right now, it's about 1,500 abstracts. Right? You go to new research at APA. It was literally 16 abstracts in a little booklet, this thin, and 4 of them were schizophrenia abstracts, and all of them were by myself and Joel Kleinman and Danny Wamberger at NIH. So the field has grown from 4 abstracts in 1976, 77 or 8, to thousands of papers that come out every year. So the field has exploded. Not too many people were involved in schizophrenia back then. I'm happy to have been involved, and many people now are in schizophrenia. So let's talk about some of the advances that have taken place. Some of them very recent, and some of them over the last 10 years. This is going to be a different kind round. It's not focusing on one topic in depth. It's going to give you like a tasting menu. You're going to hear a lot of things. It might be overwhelming, actually. So hopefully it will stimulate some residents and fellows to pursue research in a given area, the one that you like. First, the genetic advances have been incredibly exciting the last few years. As you know, a major GWAS study was done and published. And, as you know, this is the Manhattan plot showing the significant difference. This is like P less than 14 zeros at 1. This is how significant it is. And everything above the red line is highly significant. there are now, as of last week, 413 risk genes discovered for schizophrenia, spread all over the chromosomes. Every single chromosome has several of those genes. And so that's one pathway to schizophrenia. Schizophrenia is a neurodevelopmental disorder. We now have known this for 30 years. So this abnormal development of the brain can occur either with genetic pathways or environmental pathways. But as of this last month, again, the latest update is 79% of schizophrenias are genetic. And these are the genetic pathway. I just showed you the risk genes that have been discovered thanks to the collaboration of geneticists around the world. They stopped competing and started collaborating and examining 50,000 DNA samples and 50,000 healthy controls. That's where you get good findings. And then they also have found a lot of CNVs, copy number variants, which, as you know, means instead of 2 alleles, as all genes are, they find a lot of genes in schizophrenia, either 1 or 3 alleles. That means too much protein or too little protein is being produced. That will really screw up the brain development, which, by the way, the brain development, 10,000 genes are involved in brain development, which is 50% of all the genes in our chromosomes, which are roughly 22,000 right now, coding genes. Half of them are involved in brain development. So the CNVs that they found can really disrupt the effects of the other genes that are all constructing the brain at the same time. So interestingly, CNVs are pretty disruptive to the brain development. And we find them in schizophrenia, but very little, hardly any in bipolar disorder, which also has risk genes, but hardly any CNVs. And so that's another bunch of schizophrenia. At least a couple of hundred schizophrenia patients are developed because of CNVs, some due to risk genes, and then some are due to mutation. Now, the risk gene patients have a family history usually. That's how you know they carry one of those genes. CNVs and mutations do not necessarily have family history. Mutations happen in nature all the time. You having trouble hearing me?
No, we're hearing you better when you turn your head.
Oh, not when I'm looking. Yeah, that guy's good. Yeah. That's usually where I put it, but one of the many tasks of it there, I know. So there are families that never had any schizophrenia family. Suddenly they get a son or daughter with schizophrenia. That's probably due to mutations, which are very disruptive. We call them de novo nonsense mutations. And that means they make zero protein. And that can be a pretty nasty subtype of schizophrenia. So the message I'm trying to, I'll talk about genetics in a minute when I talk about the environmental factors modifying the genes. But here, just from the genetics alone, you have 800 to 1,000 types of schizophrenia, biotypes, showing you what an incredibly heterogeneous syndrome this is, a very complex syndrome. And that's a challenge for research. I mean, when you do a study on a couple of hundred patients, you think you have a great sample. Don't be surprised if somebody in Germany or Japan cannot replicate you because the composition may be different patients with different genes, with different environmental factors, which I'll come to in a minute. So this has, of course, led to a lot of biotypes and the phenotype, and I don't have to tell this department about endophenotypes because you guys have done great work. But a lot of endophenotypes have been defined both with genetics and with behavioral, clinical phenotypes. All of them are fertile areas of research, and I encourage some of the residents and fellows who are interested in research to do that. Staging is another new development in schizophrenia. Just like we stage cancer, we are now staging schizophrenia. So when patients are in the prodromal phase, that's a different brain, different stage. And they go to first episode, it's a different brain, a different stage, and so on. Second episode is a very different brain and different stage. So don't expect your patient's brain is changing continuously with this disorder, and that, of course, has a lot of implications for clinical management and for research. And the biomarkers are exploding. We have a lot of biomarkers, even more than those. That's why I just convinced Elsevier to start a new journal, Biomarkers in Neuropsychiatry, and I invited some of you to submit articles. Please do that. I'll publish it this year if you submit it within 3-4 months in the inaugural issue. But biomarkers is the way to go for precision medicine and precision psychiatry. Eventually, we're going to be able to identify who is this schizophrenia patient sitting next to, in front of me, what biotype this patient is, and what kind of genetic or non-genetic factors he or she has, and and what kind of treatment will match this particular patient. Pleiotropy has been also found in schizophrenia, which is, by the way, a common finding in medicine in general, that some genes can manifest with different disorders. The same gene, like calcium channel A1 gene. Oh, it's missing up there. The calcium channel A1 gene is associated with all those supposedly distinct disorders, according to the DSM-5 arbitrary committee designed diagnostic criteria. Actually, they all share the same gene, which tells you that eventually, as we progress more and learn more about the genetics, and pleiotropy being one of them, we might be able to reclassify the disorders in a very different way using genetic markers rather than clinical names. And another finding is that the patients with schizophrenia have a lot of family history of other psychopathology. And you can see the odds ratio here, like 400% more bipolar 1 and bulimia, and then generalized anxiety, and all those disorders that you see can occur in family members of schizophrenia. So there's a lot of overlap between psychotic disorders and non-psychotic disorders as well. Again, suggesting strongly that there are some shared genes and shared circuitry and shared neurobiological mechanisms. Now, we talked about the genetic factors. Don't forget that you can also have the schizophrenia develop from environmental influences. For instance, a lot of studies have shown that infection during pregnancy, obviously that's when the fetus is developing, can significantly increase the risk of schizophrenia in the fetus. So that's one area where you don't need genes to become a schizophrenia patient, a fetus growing up to be a schizophrenic adult. Let me go back to pregnancy here. Delivery complications, of course, hypoxia can significantly insult brain development and increase the risk. But again, during pregnancy, especially in the second trimester when the brain is developing, the 4 stages of brain development: proliferation, migration, differentiation, elimination. Each of them requires 2,000–3,000 genes. Anything that happens to a pregnant woman during that time can disrupt brain development and lead to abnormal brain, which 20 years later manifests as psychosis. Other examples of environmental causes of schizophrenia is diabetes, gestational diabetes. If a pregnant woman becomes diabetic, there's like 800% increased risk of schizophrenia in the fetus when it grows up. Severe vitamin D deficiency has been shown to quadruple the risk of schizophrenia if the pregnant female has very low serum level of vitamin D. Again, it's easy to immunize the pregnant woman, easy to manage her diabetes before she becomes pregnant, easy to supplement the vitamin D. These are manageable, unlike the genetics where we can't yet prevent them. But with those environmental causes, we can manage. We can do something about it. Famine, starving the pregnant woman can increase the risk of schizophrenia. As I said, we don't do studies to prove that. We don't starve pregnant women, but we know from the horrible Nazi era how they surrounded the Netherlands for 8 months and starved them, forcing them to surrender. Not a crumb of bread went into that country. 20 years later, there was a huge spike of schizophrenia in the Netherlands. And actually, there's recent research showing that even not just the pregnant woman, but the males during that era continue to have tremendous psychiatric and medical conditions as a consequence of that cruel act. Okay, then you have urbanicity is interesting. Many studies have shown in Europe and the US that being born and raised in a large metropolitan area of at least 1 million has 500% higher risk of schizophrenia than being born and raised in a rural area. Again, we don't know the exact reasons, but you can always speculate about pollution, infection, so stress, combination thereof, etc. Migrants tend to have higher rate of psychosis in the new country compared to the rate in their homeland. Winter births is actually 300 studies have shown that patients with schizophrenia are disproportionately born in January, February, March of the year. And we didn't know in the past why. 300 studies show that the reverse in the southern hemisphere. Now we attribute that to lack of sunshine and lack of formation of vitamin D in pregnant women, and so on. And then this is a sad environmental cause, which should be prevented, but it's not, because you keep seeing papers being published. Children who are severely physically or sexually abused in the first 5–6 years of life have a dramatic increase of psychosis in adulthood, as well as suicidality and depression and so on. It just messes them up completely. Trauma in childhood has very grave consequences, including schizophrenia. So here's another interesting development of that. It's early prevention. How early can you get? Well, how about fetal life? Well, this study was done by Bob Friedman, who was just finished his term as editor-in-chief of the American Journal. And what they did, they postulated that choline may have a protective effect on the pre-pulse inhibition, which I'm sure Niels Wardlow does a lot of every day. So what they did, they gave pregnant women, not necessarily with a history of schizophrenia, gave half of them choline during pregnancy and the other half placebo. And then they examined their children. So this is what the reason that they did the study, because schizophrenia is associated with sensory gating issues. And so they took 100 pregnant women, gave them placebo versus choline, and the infant EEG was recorded, and the normal P50 was suppressed 50% when they gave that stimulus. But the interesting point is that 76% of the choline-treated infants compared to 43% of the placebo infants suppressed the P50. So that shows you that you can actually change the neurophysiology of the fetus and protect them from potentially schizophrenia, as reflected by the neurophysiological biomarkers. And they retested them at age 3.5 years and found a similar thing, fewer attentional problems. So what's the conclusion of this? I suggest that pregnant women have one egg PID. That's a good prescription, because the yolk of the egg has a lot of choline, and that's the easy way to take it instead of pills. We talked about childhood maltreatment. I did a special issue in schizophrenia research about that. It's an enormous amount. I already covered that. But we should always ask about the history of trauma in every patient with schizophrenia to see if that may account for their illness or just additive. They may have a two-hit etiology, genetic and trauma, or genetic and pregnancy complication. It could be a more severe form of illness. And another interesting environmental cause, if you want to call it that, although it overlaps with genetic, is advanced maternal age. Several studies have shown that if men decide to conceive a baby after age 45, the risk of schizophrenia, bipolar, and autism jumps up 300%. And the reason is believed to be DNA fragmentation with advanced age, which men have to make hundreds of millions of sperm every single day, starting at age 13. And by 30 years later, I mean, you can imagine the machinery can get a little cracky, and DNA fragmentation may account for that. So it's really a kind of genetic mutation, if you like. Malaspina and others have done a lot of this work. But interestingly, the other thing I want to talk about older paternal age, they found that even the children who do not develop psychosis or autism or bipolar, they tend to have a proclivity for conspiracy theory. And these are normal people. We are all around us, our best friends. And sometimes they just believe in something that is not true. So the next time you go to a cocktail party and you're chatting over a glass of wine, and the person you're talking to says, oh yeah, 9/11 was completely orchestrated by the CIA. Just put the glass down and look at him in the eye and say, excuse me, how old was your father when you were born? Just throw him up. All right. By the way, maternal age does not affect like men, because women don't make eggs every day. They are born with 400 eggs, done in utero, so they're not susceptible to the ravages of aging. All right. Cannabis is well known to you, and there's a tremendous amount of literature showing that marijuana causes schizophrenia, triggers schizophrenia in susceptible individuals. Okay, not everybody, but how do you know? I mean, adolescents don't know. We have family history, definitely is an important factor. So it increases schizophrenia by 500%. That's why we advise everybody not to abuse marijuana during, especially adolescence. And unfortunately, Colorado and Washington State and all of those states that are happily legalized marijuana don't realize they're going to increase the risk of psychosis in kids who maybe would not have had it before. There are some genetic markers. They haven't actually nailed it yet, but there's a lot of advances finding out some biomarkers and SNPs that may predict who is susceptible. But I show this to my medical students when I lecture to them about schizophrenia. This is the gray matter loss and the marijuana use. These are healthy individuals. These are patients with schizophrenia without cannabis. They're definitely less. But worse than these are the patients with schizophrenia who are abusing cannabis. So cannabis definitely compromises the gray matter in patients with schizophrenia, which they cannot afford to do. Already low on that. So sometimes they combine cannabis and urbanicity together, confluent factors, perhaps. Recent events, and we sometimes see patients who like to attribute their psychosis to the fact that their girlfriend jilted them, or that they had a stress at work or something like that, or school. And actually it turned out to be possibly true that not only childhood adversity, but recent life events can also perhaps impose some neurobiological changes that may be different than childhood trauma, but maybe epigenetic in any case. There's also a mild encephalitis hypothesis that has been brewing in the literature. That could it be that patients with schizophrenia have a mild aseptic encephalitis that disrupts and disrupts the circuitry and leads to abnormal brain function. So this is still alive, and you know, it's not very prominent, but it's something to keep in mind as a possible etiology. And part of that actually is the blood-brain barrier permeability that may lead to schizophrenia because of oxidative stress and inflammation, neuroinflammation, and oxidative stress converge to disrupt the blood-brain barrier integrity, and that will then allow a lot of substances from the periphery to go into the brain and wreak havoc. Microbiome is becoming a hot area of research in medicine in general, and it's now emerging in schizophrenia and psychotic disorders as well as autism. That there are some abnormal bacteria species in patients with first episode compared to healthy controls, same age, matched controls. And this is still an early field, but it looks like there might be some promising leads there to the effect of the microbiota on brain development, and to be determined. Yeah, how we can intervene. They're talking about fecal transplants now in several disorders, not done yet in schizophrenia, but have been conducted in other disorders. Quinurinin hypothesis of schizophrenia, as you know, this quinurinic acid is an NMDA antagonist, and whenever you antagonize the NMDA, as we know that from the PCP literature, the 60s and 70s, when a lot of college kids used to abuse PCP and become floridly psychotic. The best schizophrenia, like drug-induced schizophrenia, is actually PCP psychosis, with positive symptoms, negative symptoms, cognitive symptoms, catatonia, thought disorder, agitation. And that's how the hypothesis, the glutamate hypothesis of schizophrenia emerged, because it wanted to know what does PCP do, and turned out to be a very powerful inhibitor of the glutamate NMDA ion channel receptor. So we now know it's a well documented hypothesis, and probably true, at least in a subtype, but it could be also a found uncommon pathway. NMDA can actually, the glutamate system controls the other receptors like dopamine and serotonin, interacts with GABA as well. So there are a lot of probably valid reasons to believe that NMDA integrity is important in schizophrenia. So we find that there's an elevated amount levels of quinurinic acid, which means that there's maybe an internal antagonist to NMDA in people who develop at least subtype of schizophrenia. Another indirect evidence also is the low serine. Serine is supposed to be an agonist to this the glycine receptor, which is a co-receptor with glutamate, and we're finding low levels of serine, which may indicate that there's hypofunction of that receptor, not due to the glutamate itself, but due to its co-receptor, the glycine. Then we, of course, have had a revolution in the last 20 years with the prodrome becoming a focus of research 1 or 2 years before the onset of psychosis. Pat McGorry in Australia is the godfather of this field, but it has spread to the US and Europe and Canada, and there are a lot of sites studying the prodrome. And of course, the hottest area there is like, can we predict who in this sample of 100 prodromal kids showing all the symptoms of the prodrome may convert to psychosis? And the conversion rate is about 35% after 1 year. So they're digging very hard to trying to find the risk factors. And the Naples group, the group here in the US led by Tyrone Cannon, have been focusing on about 115 biomarkers, but they distilled them down to about 15 with a lot of studies. And many of them are actually inflammatory. Inflammation may be the earliest phase of schizophrenia is neuroinflammation emerging during the prodrome, as measured by inflammatory biomarkers. And that's why the study by Amenger 2 years ago, where they gave omega-3 fatty acid. This is after multiple failures, by the way, of giving atypical, small doses of atypicals. Several of them were tried compared to placebo in prodromal individuals. None of them beat placebo. They all converted at the same rate. So kind of they decided to follow a different pathway, and that's where the hypothesis that could it be inflammation rather than before the onset of psychosis, there's no dopaminergic hyperactivity. So why are we giving dopamine antagonists? So that study gave omega-3 fatty acid for 8 weeks to half the patient, half the prodromal, the subject, the other half got placebo, and it beat placebo by a mile. So that was a very exciting finding. Everybody, started getting excited that the hypothesis is true. So they tried to replicate it, and NIMH funded a large study, multi-center study, which just recent last year was negative. They did not find a difference, omega-3 versus placebo. But don't jump to conclusions. Guess what the placebo was? Because of ethical reasons, they decided to give the placebo patients who were getting placebo pills CBT to help them. Well, guess what? CBT is anti-inflammatory. It's been shown in many studies that actually when you give CBT to a patient, it lowers inflammatory biomarkers. So basically that maybe erased the difference. So they're going to have to do another study where they do treatment as usual rather than a CBT, and maybe they will identify the reason there might be a truth to that study is they presented a 6-7 year follow-up of the same cohort that initially showed a difference between placebo and omega-3. And the subjects who got omega-3 were still protected 7 years later, and those who got placebo were still converting to psychosis. So it's an incredibly long-term protection, like nipping it in the bud, right? By throwing cold water on the fire, the inflammation early on in the prodrome may protect from converting to psychosis. So, it doesn't hurt to give people with a family history of teenagers with a family history of schizophrenia, omega-3 in adolescents. I encourage that. Doesn't hurt. It's a good supplement. Just in case it's true. It's safer than NSAIDs and aspirin and other anti-inflammatories, which have been given to schizophrenia, by the way. Full-fledged schizophrenia, not the prodrome, with good results. We believe that psychosis, due to multiple studies now showing that the microglia gets activated at the very early stages of psychosis. So microglia, as you know, are the immune cells of the brain. About 10% of our brain cells, entire brain cells, are microglia. And they originally were monocytes. They are white blood cells, which migrate during fetal life, cross the blood-brain barrier, still permeable in fetal life, and settle in the brain, and they become microglia. So those microglia do a good job for protecting the brain immunologically. However, when they get activated, and there are many things can activate it. It could be the biology of psychosis. It could be head injury. It could be peripheral inflammation. Once activated, they start gushing massive amounts of pro-inflammatory cytokines, interleukins, TNF, alpha, interferon gamma, CRP, the whole works, and start the process, the neurodegenerative process of schizophrenia. All right. And antipsychotics have been shown to actually inhibit microglial activation, clozapine being a good example, but other antipsychotics have also been shown to do that. And I forgot to mention to you, remember that Manhattan plot with all the genes and all the chromosomes? The highest skyscraper, which, as we call them, was the histocompatibility complex on chromosome 6 is extremely significantly different in schizophrenia patients compared to the general population. And that's the immune regulation of the body. It's clearly disrupted in schizophrenia. So there's a whole notion now that is schizophrenia an immunological disorder, not just for the brain, but for the body, which may explain why they have a lot of medical conditions. Studies have shown that young people, 18 to 20, with first episode psychosis, have a very extensive history of medical problems and hospitalization, emergency room visits for general medical conditions before the onset of psychosis. So this is a whole body disorder, not just a brain disorder. And oxidative stress, which are free radicals, okay, is well known to exist during psychotic episodes, and we can measure oxidative stress with a variety of biomarkers. And it turns out that patients with schizophrenia have an imbalance. And, unlike us, normal people make enough glutathione from the mitochondria, which is present in every cell in our body and our brain. And the mitochondria, the adequate amount of glutathione we make immediately neutralizes the free radicals that we make. Sometimes we get a little bit, not as much as patients with schizophrenia, who have tremendous amount of free radicals and very little antioxidants. There's something wrong with the mitochondria in schizophrenia. They don't make enough antioxidants, and so they are double whammy, so to speak. Very high levels, free radicals, very little antioxidants. Same thing, neuroinflammation has been shown in multiple studies to rise. All the biomarkers rise during psychosis in CSF and in serum. Again, all those biomarkers I mentioned to you, pro-inflammatory cytokines especially. And that's why when you give antipsychotics, any antipsychotic, get them out of the psychotic phase, all of those biomarkers decrease significantly. So we assume there's neuroinflammation, which is, by the way, now a well-known event in all neuropsychiatric disorders. I did a symposium a couple of years ago in St. Louis about neuroinflammation in psychiatric and neurological brain disorders. And we talked about schizophrenia and depression and bipolar anxiety and Parkinson's disease and epilepsy and sleep disorders. All of them have neuroinflammation. So there seems to be a theme in brain neuropsychiatric disorders. And that's why, by the way, back to this, about 12 studies have given anti-inflammatories, in addition to adding them to the antipsychotic, COX-2 inhibitors, omega-3, and aspirin, and minocycline, which is an antibiotic with a wonderful neuroprotective molecule, it turned out. All of them enhance the antipsychotic effect. Now, 40% of the brain is gray matter and 60% is white matter. And people talk about the gray matter mostly because that's where neurons are. But actually, the white matter is extremely important for the brain. It connects every single region of the brain together. And I don't know if you know this number, but there are 137,000 miles of white matter fibers in the brain. That's 3 times around the Earth, all inside this compact organ. And if you disrupt the white matter by compromising myelin synthesis, for instance, or through neurodegeneration with inflammation and oxidative stress, which destroy white matter, you can see what happens to the brain. You get disconnection and disconnection, 2 kinds of connection abnormality. And both can be very disruptive to the brain. And this is a picture of what the white matter looks like with using DTI. Extensive kind of infrastructure for the brain. Without the white matter, the gray matter will not work. They need to talk to each other via white matter. And unfortunately, there's a great amount of white matter pathology in schizophrenia. And turns out to be another, like neuroinflammation, and I have my students and residents working on projects to synthesize the literature. It's found in bipolar disorder and depression and OCD and anxiety and borderline personality. We're finding white matter pathology in a multitude of psychiatric disorders, but most extensively in schizophrenia. And especially the corpus callosum, which is the 200 million fibers connecting the right and left hemisphere. And I was very interested in the corpus callosum early in my career. Did a lot of studies with post-mortem samples at NIH, and actually published a paper a few years after I left UCSD about a hypothesis of how the Schneiderian delusions develop. Schneiderian delusions are, as you know, thought broadcasting, thought insertion, thought withdrawal, mate feelings, external control. And I postulated that this is before the discovery of white matter and before the advent of DTI. Diffusion tensor imaging didn't come till like 2000. So I published this in '86, postulating there's something that is disrupting the corpus callosum and causing loss of laterality, and the left hemisphere no longer recognizes the right hemisphere as part of the self with complete integration via those fibers. And so all bits of information traveling from the right hemisphere to the left hemisphere are regarded as alien, coming from outside, from some other source, some entity. So the alien intruder is actually the right hemisphere communicating with the left hemisphere, but no longer recognizable because of the corpus callosum disruption. I was very happy when the white matter literature came out showing there's extensive destruction of white matter in the corpus callosum, the biggest organ. It kind of was a validation of sorts, but to be determined. There's actually papers written about the disconnection hypothesis now that you might want to read. They're very interesting because they can explain positive symptoms, delusions, hallucinations, cognitive disturbances, negative symptoms. We can all explain them with various types of intra-hemispheric and inter-hemispheric disconnection. And there's so many types of disconnection, another heterogeneity in the equation. All right. So we already talked that. I'm going to go skip that. Mitochondria, this mitochondrial dysfunction, I already alluded to that. There's now a huge literature. Again, I did a special issue on schizophrenia research about mitochondrial dysfunction. Just read it. It's like really something wrong is going on in the mitochondria of patients with schizophrenia. And as you know, it's found in every cell in the body. They have a different genome from the regular DNA, about 2,000 genes that's in the mitochondria, separate from the nuclear genes. And they are very prone to mutations because there's oxidative phosphorylation. Mitochondria is the energy engine of the body. It's really vital. They make ATP. Without ATP, nothing works. And so because of the oxidative phosphorylation, there's a lot of potential for free radical, ROS, reactive oxygen species, which can destroy the DNA of the mitochondria. So they're very prone to mutations, and that may explain why there is some psychopathology, because children born with mitochondrial abnormalities, there are a lot of mitochondrial pathology pediatricians see, grow up to develop psychiatric disorders. Okay, but then the reverse is true. When you look at patients who are adults with primary schizophrenia, primary bipolar, or depression, you find a lot of mitochondrial pathology. The reason the brain is affected by that differentially is, even though it's 2% of body weight, the brain is 2% of body weight, it consumes 20% of all the ATP that the mitochondria produce. So if there's a compromise in the function of the mitochondria, the first organ to suffer is the brain because of lack of ATP. But then the heart and the kidney follow the brain. They also consume a lot of energy. So you get a lot of medical conditions because of that. All right. So I already covered that. Covered this. So let's move on. Medical insult. I already mentioned that to you before, that there's a lot of medical conditions at the time of diagnosis in the 900,000 individuals that were published recently from Europe. So think about it as a general immunological dysregulation of the body. All right. So these are the kind of findings they found in those young people with schizophrenia at onset, and clearly they lead to increased risk of metabolic syndrome and cardiovascular risk, cerebrovascular events, and so on. Now, here's an interesting finding about the relationship between schizophrenia and dementia, especially frontotemporal dementia and ALS. I mean, so just the last 2-3 years. So you have a relationship between schizophrenia and frontotemporal dementia that is described not in the patients with the frontotemporal who have an early onset in the 20s or 30s, sometimes present with classic schizophrenia picture. But on the other hand, there's always the family studies show that the first-degree relatives of patients with frontotemporal dementia have a higher rate of schizophrenia. So there's clearly some neurodevelopmental association between those two disorders. Maybe Krepler was right. It's a form of early dementia precox. And here's a study that came recently, 2 years ago, about the potential genetic correlation between ALS and schizophrenia. So when we talk about neurodegenerative disorders, which schizophrenia is one, it may have relationship with other neurodegenerative disorders. We should always keep our eyes open and collaborate with other specialists to get more data. Impaired glucose. We always blame the antipsychotics for this diabetes and obesity and so on and schizophrenia. Yeah, I think they contribute, but in fact, the patients have already the glucose dysregulation, as shown by a meta-analysis. Multiple studies have shown at the onset of psychosis, before you touch them with any antipsychotic, there is a high either hyperglycemia or insulin resistance or glucose intolerance, etc., all indicating that diabetes and schizophrenia actually go together, probably genetic association. And we've known for a long time that tardive dyskinesia, which by the way was my first federal grant here at UCSD, was about tardive dyskinesia. We knew even back then that patients with diabetes have a higher risk of tardive dyskinesia. If they're diabetic and receiving antipsychotics over time, their brain is more susceptible to tardive dyskinesia. Again, the literature has been growing, showing that there's interconnection between cognitive deficits, psychosis, and suicide. So patients who are cognitively impaired tend to become more high rate of psychosis and suicidality, and the reverse is true also. Patients with psychosis tend to have more cognitive deficits and more suicidality, etc. So these three together is a cluster that should be studied concurrently in a way. Here are some of the papers published about that. How about neuromotor pathology? This is really widely neglected. I don't know if you know Barbara Fish. He was incredibly productive, petite female psychiatrist at UCLA who died like a couple of years ago at 95. She did some fascinating studies in the 50s where she decided to examine the children, babies born to patients, females with schizophrenia in state hospitals back then. There's a lot of patients in those days. And, patients had sex on campus of the sane asylum, as they call them. And they used to take care of the babies. So she decided to study them and follow them up for 25, 30 years, passing the age of risk, and see what is the difference between those who become schizophrenic and those who don't, early on. So she started observing them on the first day of life and took notes, and then did neurological exams on those babies and followed them up, and found out later who became and who did not become schizophrenic, and went back to her notes. And she found that on the first day of life, believe it or not, the first day of life in the newborn nursery, there was already a difference between the babies who later become schizophrenic and those who don't. So how can that be? First day of life? Well, she found out that they are awake an extra hour. Okay, so newborn babies sleep 23 hours a day. Wake up 3-4 times, suckle some milk, go back to sleep. 23 hours, they sleep. Those who became schizophrenic, she noted, very good observer, were awake, eyes wide open for an extra hour in the first day of life. What does that tell you? Their brain is hypervigilant. It's watchful. Their CNS, that physiologically is alert. So you can stretch it and call it, this is the earliest manifestation of paranoia in a way. So those newborn babies don't talk, but if they did speak, they probably accuse the nurse of stealing their milk or something like that. I'm just kidding. But that shows you that they're physiologically already different at first day of life. And the brain, you don't have to wait 20 years to see a patient develop psychosis, say, oh yeah, they have schizophrenia. Manifest throughout the life cycle. And then part of the reason I mentioned that is because she examined them neurologically, found that they had a lot of neurological soft signs, very erratic developmental milestones. They would achieve a milestone and lose it instead of going forward to the next milestone. And she noticed that they have very flaccid kind of muscle tone, and they walked about 3 months later. Delayed walking was very common in the kids who became schizophrenic. There are a lot of motoric abnormalities in patients with schizophrenia, but very few people pay attention to them. Generally, the clinicians, when they get a patient with acute psychosis, the last thing they do is test their muscular function, their motor function. They just quickly give them antipsychotic, and then they blame all the motor abnormalities on the antipsychotic iatrogenic effect. But there's actually a lot of motor pathology in schizophrenia that that tells you the neuromotor region of the brain is also impacted. A review of the literature actually showed that patients at first episode, never medicated, drug-naive patients. Look at the Parkinsonism. This is the percent of patients who have Parkinsonism, rigidity, stiffness. Not necessarily the classic picture of Parkinsonism, but rigidity being the most common. And then some of them have dyskinetic movements. Some of them had dystonias. And overall, EPS, extrapyramidal motor abnormalities, are quite common in schizophrenia. Again, because it's a heterogeneous disorder, it shows you that there is a subtype that has extensive rigidity and dystonia and dyskinesia. Others don't. So again, you can't generalize, but you have to realize that there are subcategories. The eye is the window to the brain, right? And in the last few years, literally the last 5 years or so, a lot of studies are showing that you can see abnormalities of the retina in schizophrenia, really distinct abnormalities. One of my residents did the review of the literature about that. But it's also becoming very hot in Alzheimer's. They're finding that there are some early manifestations in the retina that can predict who is going to become an Alzheimer's patient long before the onset of cognitive deficit. So this is another area of research that's worth doing. And fortunately, we have now a lot of techniques to measure retinal function, structure and function that can be employed here. We should collaborate with ophthalmology. Some of the residents and fellows might want to do a study and look at other populations, schizophrenia and bipolar and depression. Maybe you'll find something interesting. All right. And the microvessels, by the way, seem to be quite abnormal in patients with schizophrenia. The vasculature of the retina, perhaps in the rest of the brain, seems to be abnormal. Trying to find out exactly how does this relate to psychosis, but it's a finding. Many of you know what melatonin is, and some of you may use it for jet lag or for sleep at night, or whatever. But actually, melatonin can attenuate metabolic syndrome. There are several papers showing that melatonin and melatonin agonists can actually counteract the antipsychotic-induced obesity and metabolic dysregulation. So this is, again, not well known, recognized as potential treatment for patients who do oftentimes get obese and diabetic. What about relationship with autism? There is a lot of overlap, genetic overlap, but they're really two distinct disorders. But because they are spectra, everything in psychiatry is a spectrum. Spectrum of schizophrenia, spectrum of depression, spectrum of autism, and they overlap. There are areas of overlap. One example of genetic overlap is a gene on chromosome 16 that was recognized and published in Nature a few years ago, where this gene actually controls brain growth. It inhibits brain from growing excessively, so it kind of fits inside the cranium, right? So it's inhibitory. Well, they found that patients who have one copy, which means they're less inhibition, grow much more than necessary, and they become autistic, which autism is associated with a 50% of the increased brain volume. Doesn't mean it's a better brain. It's cluttered with cells that were not eliminated. So actually, the highways of the brain are not functioning very well, leads to disability. And the subjects who had 3 copies of that same gene become either schizophrenic or bipolar. Too much inhibition, so they're born with a brain that's 10% smaller, roughly, which is what we find in babies, newborn babies who become schizophrenic. They're born with smaller brains already in fetal life. So that's an example of how one gene can cause either, depending on how many copies you have, lead to one way or the other. Now, who wants to give rat poison to patients with schizophrenia? Okay. Warfarin is a blood thinner, and a recent study showed that you get a full remission with Warfarin IV. Now, this was not done on the lark or a whim. There's actually some evidence that there's some coagulopathy in patients with schizophrenia, another abnormality that we didn't know about. And perhaps anticoagulants, and this is just one of several papers, may help the symptoms of schizophrenia. So to be replicated before you adopt it, because it's high risk, obviously. But it's an interesting clue to what's going on in terms of the blood thinning in schizophrenia. Telomeres are a way to recognize your cellular senescence, how old you are, and how much you're aging. And as expected, patients with schizophrenia, Philip actually did a special issue for schizophrenia research on accelerated aging in schizophrenia. There's a lot of reasons to believe that because there's a lot of literature. And now the telomeres are the end of the chromosome. You know, they look like the things on your shoelaces. You know what they're called? Amazing. I found not too many people know those little plastic things at the end of your shoelaces. They're called aglets. I didn't know that until I looked it up. So the telomeres are the aglets of the chromosome. And with every cell division, these telomeres shrink, and so it predicts aging, and we find that they're actually accelerated telomere shortening in patients with schizophrenia. And they're done with the leukocyte in the blood, and they clearly correlate with accelerated aging, which I wrote an editorial about that. Digging in the literature, I found a role for the mitochondrial dysfunction, inflammation, oxidative stress. All of them tend to contribute to telomere shortening. So it's an interaction of multiple pathways that exist in schizophrenia and compromise their living longer. They die about 20, 25 years before their time. So, articles like this have been coming up in the literature. And Philip, are you still studying accelerated aging? Okay, so keep it up, please. Give us more data so we can establish the link with schizophrenia. And it's related. Accelerated aging, this is a model of how inflammation and oxidative stress eventually compromise protein carbylation and lipid peroxidation and DNA hydroxylation, etc., and lead to the accelerated aging. Now, here's a pharmacological finding. I don't know how many of the residents. Residents, can you raise your hand? I want to see. Not too many of you are here. Okay. Oh, good. There's some online. That's good to know. Now, Haldol, I used Haldol in the 80s. It was like the best antipsychotic because it works so well. No doubt it works very well. Unfortunately, in the last 15 years, we have 31 studies, individual studies, published in neuroscience journals, which clinicians rarely read, showing that haloperidol kills neurons in 15 different molecular mechanisms, which I'll show you here. So I actually decided to review that literature and put it in a clinical journal so that clinicians can read it. And these are, every one of those bullets is a mechanism of neurotoxicity and neuronal cell death. So patients who are yeah, it works on their psychosis, but if you keep them on it, their brain continues to atrophy and shrink. They're losing great amount of cells, neuronal cells, which the atypicals don't do at all. I'm coming to that later. So please stop using the conventional antipsychotics. Many of those studies included other antipsychotics like promethazine, fluphenazine, and perfenazine, the drug that was in the Katie study. It's just as bad. But Haldol is the one that all the researchers have used to replicate one another. So please try not to use this old 50-year-old antipsychotic because it's dangerous to the brain. It works, but at a heavy price. On the other hand, atypicals have 12 different mechanisms that actually promote brain integrity and protects the brain. Again, we just published this paper a few weeks ago. Again, one of my residents published this, reviewing all the literature. And here are examples of what atypicals do to enhance brain integrity and protect the brain, neuroprotective. They enhance neurogenesis, which is critical to replenish the brain. Increase neurotropic factors like BDNF and NGF, NGF, the neurotropic factors should increase. You know what Haldol does when you give it to a patient, to a rat? 50% drop in the NGF. It does the same thing in humans. On the other hand, if you give any of the atypicals, actually increases the low. NGF and BDNF are 50% low in psychosis. So Haldol makes it worse. Atypicals increase it, go back up. Reversing gene expression by PCP, which is a nasty psychotogenic drug like we talked about. Even stroke, I mean, we don't use atypicals for stroke patients, but neuroscientists want to do that in rats and see what's the effect on stroke. So they ligate the carotid and get a stroke, and you get massive damage, ischemia in the brain. If you pre-treat with haloperidol and then ligate the carotid, the damage is just as bad. If you give the rat a couple of weeks of an atypical and then ligate the carotid, the ischemia is mild, far, far less than it is without an atypical. So it shows you that there are some neuroprotective effects that may not be clinically relevant to us, but it tells us something about the atypical. Here's another interesting thing. Dendrites have spines. Yes, they have numerous spines. As we speak right now, I've been talking to you for an hour, and you formed hundreds of billions of spines in the visual area, in the auditory area, in the limbic system, because I stimulated all those regions in your brain. So it's experiential neuroplasticity. That's the wonders of the human brain. You're going to leave this place with a completely different brain than the one you entered, but your lungs and thyroid and bones and spleen are exactly the same. Okay? The only organ that changes is the brain. So one of those immediate neuroplasticities are spines. They look like little mushrooms, grapes coming out of dendrites. I wish I had the picture with me. Maybe I do have it. No, I forgot to put it in. So dendrites have a lot of spines that form from every single day of your life. You're learning. You're basically developing a spine. Post-mortem patients with schizophrenia, naked dendrites. There's hardly any spine. Psychosis depletes all the spines, wipes them out. So that's very hard for patients with schizophrenia to learn new things. There's no learning. They cannot form spines that immediately encode the biological basis of new material that you learn. When you give PCP to a rat, it wipes out the spines, exactly like psychosis, which, by the way, it causes psychosis in humans. So maybe that's another example of how PCP can cause psychosis. No spines, gone. So what atypicals do is an interesting study in rats. They give a rat PCP, look at its brain, all the spines are gone. They pre-treat with haloperidol, and then give PCP, and the spines are gone. Haloperidol does not protect it at all. You give an atypical, they use 2 or 3, I'm not going to mention their names, but they're well-known atypicals, and they give PCP, and then look at the brain, the spines are intact. It protects the brain from losing its spine. That's a neuroprotective mechanism, which is probably happening in our patients. It's good to know that. And so on. They also prevent the damage to oligodendrocytes. These are oligodendrocytes are the glial cells that manufacture myelin every day. You know how important white matter is? I already talked about it. So it's a way to restore the white matter integrity, so as to speak. And so on. There's a lot of other mechanisms that you can read there. And read the paper. We just published this in schizophrenia research. All right. What's the mechanism? Why are the atypicals different? It turns out that if you block dopamine very harshly, like the old antipsychotic, haloperidol being an example, 1,000% higher dopamine antagonism than serotonin 2A, you actually decrease the neurotropic factor BDNF, which has a lot of implications. But if you block serotonin 2A, much more than dopamine D2, which is what all atypicals were designed to do. They were modeled after clozapine, so 500 to 800% stronger antagonism of the 2A receptor of serotonin, and compared to the blockade of dopamine, then you increase BDNF. So that may be the real reason why the class of atypicals are better. All right. Here's another finding that's worth mentioning for clinical management. I'm very fond of injectable antipsychotics, but I like to use them as early as possible, not wait for several episodes. Every psychotic episode destroys the brain. Roughly, this is the number in several studies, brain loses about 12 cc's of tissue with the first episode, another 11, 12 cc with the second, another 11, 12 cc with the third. Pretty soon the patients have lost 30, 40, 50 cc's if you allow them to relapse, mainly due to poor adherence, which is very difficult for patients to do for many reasons. Patients with schizophrenia cannot adhere to medication. Many of them don't even believe they need the drug. Lack of insight, negative symptoms, they don't have the motivation, cognitive deficit, they have severe memory deficit, they forget. So, and then drug abuse, which makes it impossible for them to take drug when they're stoned on drugs. So our patients keep relapsing and losing brain tissue and become disabled. You cannot lose that much brain tissue and recover. That's why they all look disabled, and people think schizophrenia is a devastating brain disease that's it's untreatable, and you're going to be disabled the rest of your life. Because you look around you and everybody seems to be that way. But that's because we're allowing them to relapse again and again. Imagine if you can give the injectable and ensure adherence at the very beginning, after the first episode, and never let them have another episode, how much better that would be. So this study was done by UCLA, actually. Is the paper there? Yeah, Sabatnik and colleagues, and Keith Nuchterlein is the senior author there. They actually gave half the patients injectable atypical, half oral atypical in the hospital, discharged them, doing well. First episode respond very well, better than any other future episode, rather. And for a whole year, this group got the prescription refill, this group got the injectable, right? What did they find at the end of the year? What I just showed you, 650% lower relapse rate. I mean, this is amazing. If you can protect your patient from relapse, you've got it made. Your patient will be protected from further neurodegeneration, and the outcome will be much, much better. In fact, Robin Emsley in South Africa, who's a very, very well-known researcher, did the first study of injectable antipsychotics in first episode patients, and in 2006. A 2-year study, patients signed consent form, he got them from the community. And what did he report at the end of 2 years? 65% were completely back to normal. Went to school, back to work, functioned great, not a single relapse for a full 2 years. That's great, 65% in a very heterogeneous syndrome. It's remarkable, right? And they got their insight back after several months, 8, 9 months. You know, they really were very happy with their treatment. So the study finished. He referred them back to their referring psychiatrist. Every single one of them was put back on pills, as if the study was just like just a study, not meaningful. 100% of them relapsed within a few weeks because they cannot take the pill. So sad, right? But a very important insight came out of that study, unexpected insight. I asked Robert, I said, did those psychiatrists see the light and put the patients back on the injectable that kept them well for 2 full years? He said, oh yeah, they did. But, and that was the unexpected insight, 18% of the patients, which is 1 in 6, never responded again. They developed complete treatment resistance to the same drug that kept them well for 2 full years. That's how quickly treatment resistance develops. After only a second episode, there you go, 1 in 6 patients. Imagine how many more get it with subsequent episodes. The brain is changing completely. It's no longer the same brain. Psychosis disrupts the receptors, disrupts the connections. It's no longer the same neurochemical landscape. And no wonder patients, subgroup develop it very quickly, another later and later and later. Other studies show that you get about 14, 15% treatment resistance with every episode. So it's really important. If you want to manage your patient, that's how I would manage a relative. My family member becomes psychotic, I would give them long-acting immediately, knowing what I do. And I just mentioned a couple of studies, but there's a lot of data that supports that. Minocycline is, I mentioned it earlier as an anti-inflammatory, but actually there are a lot of studies, many of them coming from Japan and China, about minocycline in schizophrenia. If you add it to the antipsychotic, it seems to really accelerate antipsychotic effect. It's a very good neuroprotective agent. I don't know how many of you have used it, but it reduces glutamate excitotoxicity. It's anti-inflammatory, it's antioxidant, it's neuroprotective. And so this minocycline, I think people are recognizing it could have a role in the treatment of schizophrenia, which is now not a standard of care. Just several papers, at least 20 papers published. This is a finding that bit the dust very recently. Intravenous nitroprusside, which is an antihypertensive drug, given IV, reported by a group in Brazil, complete remission. Well, everybody got so excited. It was a small sample, 20 patients. And the replication just came out last month showing it does not work. It shows you the hazards of jumping to conclusions in schizophrenia research. But it looked good for a while because nitroprusside actually is an NMDA agonist, so it theoretically should help schizophrenia. This is the paper that was by Halleck and colleagues. Rapid improvement with intravenous nitroprusside. Unfortunately, didn't get replicated. Clozapine causes low mortality, including suicide and cardiometabolic. This is really paradoxical because clozapine causes a lot of weight gain and diabetes. Right? You would think that patients would die from heart disease, stroke, and whatnot. But Tihonen in Finland, who is the chief of psychiatry there, followed up patients for 11 years and found that actually clozapine has a 11 years lower mortality than all the other antipsychotics. That was a study a few years ago. But more recently, he actually found that long-acting injectables in Sweden, they did a study about mortality and found that the patients who were taking long-acting atypical injectable, they named it specifically, had the lowest mortality after 7 years than all the others. And the highest mortality, 800% more mortality, was with Prolixin and IM, which is widely used as a injectable, but it was harmful to the brain. Estrogen is a neuroprotective agent. It's actually an antipsychotic. And since 2002, especially with Dr. Kalkarani in Australia, a lot of studies have been done showing that if you add estrogen, especially raloxifene, to females, initially they started with females. If you add it to females who are not responding to the antipsychotic, it actually potentiates the antipsychotic effect with good results. And they first gave it as an adjunctive therapy, and then they started using it monotherapy in females, good results, and then they started giving it to males. And they were initially afraid of feminizing the males, but actually it did not happen, and they got very good results in the male patients who were not responding sufficiently to the antipsychotic. N-acetylcysteine is a powerful antioxidant. It's a supplement in the health food store, like omega-3. It's not approved by the FDA. But this is one way you can help your patient mitigate the damage to the brain from oxidative stress, which we mentioned earlier, occurs during psychosis. So several studies have added N-acetylcysteine, NAC, we call it, to the antipsychotic and got a better outcome, especially the first 1 or 2 episodes. It becomes a little less effective with chronicity and brain damage already too late at that point. But using it early is good. But unfortunately, patients will have to pay for it because it's not covered by insurance. NAC becomes glutathione in the body. So that is a way to replenish the antioxidant. Okay. And here's one of many studies that have been done for N-acetylcysteine. Can I now talk about confusion? We know that cannabis causes psychosis, right? It triggers psychosis, we mentioned earlier, but a subset within the cannabis, which is cannabidiol, actually has been shown to improve psychosis. It's a very thin sliver of the patients who actually seem to respond. We don't know who they are yet. We're trying to find out a biomarker that identifies those who should smoke marijuana. But it's like a very small fraction of patients. All in all, it's like tobacco. I would avoid it. All right. And tardive dyskinesia, which is how I started my research at UCSD, we became totally hopeless that it's never going to be treated. I tried multiple drugs, published 15 papers about PD, all of them negative study. Finally threw up our hands and said, it's not treatable, forget it. PD is unmanageable. So you can imagine how happy I was when in the last couple of years, 2 different companies introduced 2 different drugs, both the same mechanism of action, which is the vesicular monoamine transporter number 2 in the brain, that actually were approved by the FDA for tardive dyskinesia. They work. I use them in my patients. So this is a very major advance. If you have patients with PD, you should try one of those 2. They both work. Mortality, I mentioned earlier. Here's the 7-year prospective study that was published in Schizophrenia Research by Tapali and Tehonen, that group. 7-year study looking at mortality and found that this particular atypical injectable paliperidone palmitate had the lowest mortality than all the others, oral or injectable, typical or atypical. But the atypicals in general had much lower mortality than the typicals. But the injectable, delivering the drug was clearly helpful to lowering mortality. You know what was the worst mortality in that study? No antipsychotic. The patients who were not receiving an antipsychotic had the highest mortality, which shows you how useful these drugs are, contrary to the idiots like Scientology who say that we are harming our patients by giving them antipsychotics. Non-dopamine, this is an interesting new finding. I just published this finding 3-4 weeks ago. A non-dopaminergic antipsychotic, which is pimavanserin, it doesn't block dopamine at all. It just blocks, it is a powerful inhibitor of 2A, but we call it inverse agonist because it's even stronger than regular antagonist. Pimavanserin, which is called Nuplazid, approved by the FDA for Parkinson's disease psychosis, which affects 50% of patients with Parkinson's develop hallucination delusion. And this has turned out to be an ideal drug because it doesn't worsen the motor symptom, doesn't block dopamine, it's already compromised, and it helps reduce the psychosis. So I thought to myself, I have patients who have failed clozapine. What do I do with those patients? They were already refractory. They failed multiple antipsychotics. I gave them clozapine, adequate dose for several months, still hallucinating and delusional. What do I do? You know, you got to do something to help those really miserable patients. 50% of clozapine patients are treatment resistant to clozapine. So I decided to use this drug, pimavanserin, saying, hey, it works in Parkinson's disease psychosis. Let's give it a try. And it did. It worked very well in clozapine refractory patients. After only 4 weeks, we gave them the same dose as Parkinson's disease psychosis, 34 milligrams a day, and published the paper. We had 10 consecutive patients who initially we added it to clozapine. It worked very well, 6 patients. Then we said, why not give it before clozapine and avoid all the side effects of clozapine? And it worked in 4 more patients. So it's a hypothesis-generating finding. People got excited about it, actually. Like many of my colleagues sent me emails telling me they're very happy to see this. And we're now going to subject it to a controlled study to see if we can actually use it in lieu of clozapine, or in patients who refuse clozapine because they don't want blood draws, or in patients who develop severe side effects, sometimes fatal almost. It's almost done. Unmet needs. Unfortunately, we still have no treatment for cognitive deficit and no treatment for negative symptoms of schizophrenia. So there's a Nobel Prize waiting for one of the residents if we want to go and try to find something that works. Because many attempts have been made. Still no treatment inside. This is the cognition of patients with schizophrenia compared to the general population. Here, the spectrum, and patients that are shift to the left. So most of them, but not all, some of them have good cognition, but the majority have 1 to 2 standard deviation below the controls. And the negative symptoms, as you know, are all disabling to function, and we still don't have any treatment, although multiple attempts have been made. I think I talked to a couple of my colleagues yesterday trying to do CBT and social skill training. Some might help some of those. Why not? Cognitive remediation also is very promising, but it's not widely used. Just a few psychologists are using it. It hasn't penetrated clinical practice yet. So in conclusion, numerous etiological factors, genetic, environmental, numerous neurobiological abnormalities, many pharmacotherapeutic strategies, both dopaminergic and non-dopaminergic. A lot of unmet needs remain, and we can and should improve patients' physical health and mortality. Thank you very much for your attention. Thank you. I know I overwhelmed you with a lot of findings, but you know, it's supposed to stimulate you, and hopefully you will pursue one or more of those that you know that you're interested in. Igor?
First of all, Henry, thank you so much for this tour de force. Very much appreciated. One thing I should have warned you about is that our grand rounds are actually not only focused on trainees. You're mostly seeing faculty.
Of course, well, I love trainees.
But they can learn something too.
Yeah, we're going to influence them early, and they will become researchers. That's my goal, is to bring them to academia. The faculty are already sold on it.
Right. So our tradition here is we allow one question because everyone likes to ask multiple questions, including my friend, Dr. Barbara Perry. I'm limiting her to one question. She's usually the first one to ask a question, Barbara.
Well, thank you for this very comprehensive presentation. Of course, one of the protective factors of schizophrenia is being female, and I'm just wondering, you know, estradiol, as Neil informs me, can affect P50 gating. It can affect kynurin. Can affect dopamine, serotonin, melatonin. There's increased dendritic spines. There's a bigger callosum. Corpus. Which is the relevant factor? Would you say? What are your thoughts?
What does it?
Why is it the women have a protective?
I wish I knew the exact mechanism, but you know, you're absolutely right. It's neuroprotective in multiple ways. It enhances the brain structure and subsequently function with all the things you just mentioned. But females are so lucky. They're born with an antipsychotic, right? And the estrogen helps you, and that explains why there's a later onset in patients with female patients. They're about 10 years shift to later, which helps them a lot. They go to school, they get a job, they may get married even before the onset of psychosis at 25 to 35, whereas males get it from 15 to 25. And to make things worse, males actually have a psychotogenic hormone called testosterone. So not only that, it is psychotogenic in contrast to estrogen. So until we find out how it works, I think we should pursue it as a potentially useful adjunctive therapy. That's all I can say at this time.
Are there other questions, comments? Yes, Dilip. One question.
Thank you, Henry, for a great talk.
Thank you.
I was very interested in your non-dopaminergic
Yes.
antipsychotics. Do you see that as the future of psychopharmacology?
I see that as a very good alternative to anti-dopaminergic drugs in a subset of patients. I really, and I wrote that in the paper. I think that the clozapine refractory patients may be a serotonergic psychosis, which is different than many of the other patients. The patients who respond, the 65% who respond to the first antipsychotic in the first episode, are probably dopaminergic psychosis. And having the atypicals block serotonin 2A along with the dopamine may help a little bit. But even Haldol, which is a very strong dopamine antagonist, helps a lot of those patients with detrimental effect. So I think that it is a clue that there is, in this heterogeneous syndrome, there is a subset that is a serotonergic psychosis, not dopaminergic psychosis. That's why, throwing a lot of anti-dopamine drugs at them will not work. Yeah. And in fact, I like to use injectable antipsychotics in the first episode, because if the patient doesn't respond to guaranteed adherence for 6 months, I know that I've identified the potential clozapine candidates or this new candidate. We can catch it early rather than wait for several years and try multiple drugs.
I wonder if there's any data on the use of glyphosins in diabetics, the glucose reuptake inhibitors in patients that have schizophrenia also.
Yeah.
The glyphosins reduce cardiovascular mortality, surprisingly, probably because of reduces inflammation. I wonder if there's any emerging data on patients that are maybe schizophrenic and diabetic and are using these drugs, and maybe they improve more.
Using which drugs?
The glyphosins, empagliflozin, dapagliflozin.
Oh, the hypoglycemic agent?
Yes, the
Yeah.
glucose reuptake inhibitor.
Yes. To my knowledge, there have been no studies of using hypoglycemic agents, various types, different mechanisms in schizophrenia. But that's a very interesting hypothesis, and it's worth testing. The question is, where do you test it? For ethical reasons, do you do it in the first episode or later? And if you use it later, it means there's already damage done, so it may not be representative. That's the dilemma we find ourselves in. I mean, one of the studies I would like to do, which I cannot do because I'm sure human subjects will not approve it, is to give 1,000 milligrams of prednisone to IV to patients with schizophrenia, just like my colleagues in multiple sclerosis, MS, do with a relapse of MS. They give them 1,000 milligrams IV, a huge dose. I would like to see what that does to schizophrenia, because theoretically it might work, but it's hard to do theoretically. So you have to take that first step, but taking it is difficult. Same thing with the question.
Any questions back here? Comments?
Yes, sir.
Oh, there we go.
I have another question on pimavanserin. Am I saying that right? The new serotonergic antipsychotic.
Yes.
What about the metabolic side effects with this mechanism? Are they different from what you would see with a more traditional D2 blocker?
Yeah, amazingly, very little. If you look at the FDA study with Parkinsonian patients, it was only 6 weeks, but they have a lot of patients who stayed on it for a whole year, open label. There was no metabolic dysregulation. So it's different for some reason. It is safer, but it's also safer with all the other side effects of clozapine, like I guess hypertension and excessive sedation and sialorrhea and cardiomyopathy and seizure risk and a whole mess, side effects of clozapine are avoided with this drug. It has like a few side effects, 3, 4, 5% of the patients have side effects. Paper by Cummings, Jeffrey Cummings summarizes that. It was the FDA study.
Dr. Strong, do you know if there's a plan to extend the indications for pimavanserin to other conditions other than Parkinson's psychosis?
I think the company is likely to do that. I did this without the knowledge of the company. I did it on my own, and when I published it, the company called me. They were delighted, and they are actually conducting. Yeah, I mean, they see another opportunity. They're doing a study for depression, by the way, and for Alzheimer's right now, conducting an FDA study for treatment-resistant depression, adding pimavanserin, because it has a signal to improve depression as well, and in the psychosis of Alzheimer's, for which nothing is approved. and they believe this might actually, they have an early signal that it might actually work, but we won't know the full results for another year. So yeah, they're pursuing it, and they probably were going to embark on a schizophrenia study as well. You have a question?
Just a very practical question.
Yes.
In terms of the typical antipsychotics, what are you doing in your program with the residents and your attendings in terms of, are they being utilized at all?
Which drug?
The typical antipsychotics?
I make sure that all the residents, my residents, know the facts from day one of their first year of residency, and they all use atypicals. Once in a while, they have patients who come in with on one of the first generation drugs, and a patient refuses to change. So some of our patients don't want to change in the clinic. So they educate them and try to convince them. But other than that, they use atypicals. There's 11 atypicals on the market, plus clozapine. Why do we have to use the first generation drug that was introduced in 1965? That year, 1965, when clozapine came, when Haldol came to the market, was a huge big deal, very different than phenothiazines, and people adopted it. That same year, Reserpine was the #1 antihypertensive in the United States. Anybody using Reserpine nowadays for hypertension? They would laugh you out of your profession if you do that. There are 6 generations of antihypertensive. We're stuck using the first generation, despite the fact that it's harmful, which we didn't know before. I used it because it was the state of the art back in the 80s, 70s, and 80s and 90s. Now that we know better, we should really avoid it. But you go to jails and prisons in the U.S., probably here in California, and they use Haldol right and left because it's cheap, and those criminals don't deserve any better. They criminalize the mentally ill, they treat them like trash, and they give them the cheapest possible drug, even though it's harmful to their brain. And I contacted the former FDA, Psychiatry FDA head of pharmacology, and got his advice about how can I convince the FDA to pull it out of the market, or to put a huge black box on it, based on the published findings. And he said it will take a while, but it's eventually might happen. It takes several years. It's hard to do.
Thank you. I know the department would be disappointed if we didn't conclude with a question about cannabis, because
okay.
We have a Center for Medicinal Cannabis Research, as you may know. So square a couple of things with me. Clearly, there's an association of psychosis onset and cannabis use. I think that's been established. And yet, the overall prevalence of schizophrenia in the world hasn't changed, as far as I know, despite introduction of cannabis in the 60s. How do we square that? Is this merely a brought forward in time phenomenon, or do you believe that there is really a significant risk for de novo chronic schizophrenia with cannabis?
Yes.
Just an easy question.
That's a good point. I think that the reason that this incident, rather the prevalence of schizophrenia, has been stable for all those years, and will remain stable despite the lack of procreation. Most of the males never have sex, never have children. A few females get married before they have it, so maybe they perpetuate something. But it's the environmental factors play a role, and the genetic factors play a role, and together they will maintain that roughly 1% around the world, even though I should remind you that it's 1.5% in the northern hemisphere where there's very little sunlight. Vitamin D plays a role in the very northern part of the world. And around the equator, it's 0.5%. Equatorial countries with a lot of sunshine have 3 times less, but overall the incidence is 1%. I don't know whether do you have data that marijuana is actually cannabis is significantly more now than it was 30 years ago or 60 years ago? People have been using marijuana all along. I don't know that anybody has actually looked at the actual usage, which is hard to estimate.
No, I was just saying, again, we don't want to have just a marijuana symposium here, but if you look, I mean, we've had epidemiologic data on schizophrenia for many decades,
Correct.
Including decades preceding the 60s and 70s,
Correct.
when marijuana use became prevalent.
Correct.
And it seems like, looking at those data, there isn't much change.
Correct.
Over 7 decades even. So anyway, it's kind of curious. And the other is knowing that even THC has potentially neuroprotective effects. What about adding marijuana to Haldol and saving the...
This guy killed the patient. Two hit the structure. I will tell this for the sake of the residents again. That how do you know if the patient psychosis was caused by marijuana abuse, or were they going to be schizophrenia patients anyway, and marijuana was just, and the last straw, so to speak. Well, a convenient clinical way to identify the at-risk the psychosis, cannabis-induced psychosis, versus schizophrenia patients who happen to smoke and were really going to become schizophrenia anyway, is to look at cognition. I mean, cognitive function to me is even more useful than an MRI. If you do cognitive function in an adolescent and you find it's already on the lower end of the spectrum, like one standard deviation below normal, and they're abusing marijuana, they probably were going to develop schizophrenia anyway, or they're more likely to, compared to somebody with a very good cognitive function. In fact, normal, and using marijuana, they're probably not at risk. It's like a little clinical clue because the cognitive deficits actually precede the onset of psychosis.
So one wrap up, and then we have to quit.
I had a question, and again, your talk was amazing. So if a patient had not tried marijuana before the onset of schizophrenia, could they possibly be a part of that subset where smoking marijuana may help them, or using THC therapies?
Actually, the contrary. I've had patients who swear to me that marijuana helps them, and that's why they're smoking it. And they come in with psychosis, but marijuana helps their anxiety, their paranoid feeling. It relaxes them. So if anything, they stumble on marijuana and they self-medicate as a way to improve their psychosis, or the incipient psychosis, actually, at least. But yeah, it's hard to tell who are benefiting and who are not. I regard it, since 95% or more is going to be harmed, I'd rather stop using until we find out who are those very small percent whose biology actually responds better to cannabis. Cannabidiol, actually, not cannabis.
Well, we do have to wrap it up. Thank you so much, Henry, for first of all coming here.
Thank you.
For endowing this enduring lectureship, which will occur every year. And you have inaugurated this in a gangbuster fashion.
Can I take a picture with you before I go?
Well, let's have somebody else take a picture.
Yes. No, I won't take a picture of you, just to remember the moment.
Before you take the picture, I have to do our presentation, which is your UC San Diego School of Medicine, Department of Psychiatry hat. Wear it proudly, even as chair of your department.
I will.
And especially when you go out into the sunshine of St. Louis or wherever you go.
Thank you. All right. Thank you.
You have to put it on something.
Thank you. And